Good evening, and thanks for coming on this cold spring evening. My name is Steve O'Rahilly, and I'm a researcher at the university here, where I direct the Medical Research Council's Metabolic Disease Unit and co-direct the Institute of Metabolic Science. I'm also a practicing physician at Addenbrooke's Hospital, where I look after people with various unusual forms of diabetes. And by training and instinct, I'm a clinical endocrinologist. I've always liked hormones and looking after people whose hormones go wrong. And the hormone that really got me hooked some 35 years ago was insulin. And that's a hormone that controls blood glucose as one of its major functions. Obesity, however, is a topic that both professionally and personally I've wandered into rather by accident. It is, however, it turned out to be a rather fascinating topic, linked, of course, because many obese people do develop a particular form of diabetes. But of course, you can have many types of talk on obesity. You could have a cosmetic talk on obesity. You could have a sociological talk on obesity. You could have a moralistic talk on obesity. You could have an economic talk on obesity. So you're not going to get much of any of those today, because I am both a biologist and a scientist on one hand and a clinician and practitioner on the other. And my approach to telling you about the science of fat today is unashamedly biomedical. From a biological point of view, I believe we share a lot of our biology with other creatures that we share the planet with, and I'll talk to you quite a bit about how they and we regulate our body weight. And as a clinician, I'm particularly interested in how we can practically improve the health and lives of people with conditions that shorten them or make them more miserable. So they're the two kind of perspectives I will try and take today. And to start with, I'm going to really tell you, from the point of view of your biology, how you might avoid getting type 2 diabetes. Well, the first thing you might do to avoid getting type 2 diabetes is to maintain what we call energy balance. Now, energy balance has a specific meaning. It means that over a particular period of time, which you define, let's say a month, the amount of energy you take into your body in calories, the amount of calories, is balanced by the amount that you expend either sitting around, the amount you burn, or how much you burn by exercise or fidgeting or whatever. And that energy, if you're in perfect energy balance, you will weigh not 1 gram more at the end of the month than you would at the beginning of the month. So a good way of avoiding getting diabetes is to remain in perfect energy balance. It's hard to do as we get older. But if we don't remain in perfect energy balance, we're fine, because we have a professional tissue in which has been designed to store the excess energy that we take during periods of our excess. And that professional tissue is called adipose tissue or fat tissue. It's not there just to be annoying. It's there for a genuine biological reason, to store energy during periods of feast, so that we can keep our energy stores when we're infected or our food is we're deprived of food. And so it's a very highly sophisticated professional tissue that I'll introduce you to as we go through the talk. But, you know, if you start being unable to store that energy safely in fat, you tend to put it in other places. And the places it goes to are places where it causes mischief. And if that excess energy is going into, for example, muscle and liver, there it can cause real disruption, particularly in the form of fat in those tissues, real disruption of how insulin controls blood glucose through those tissues. But as long as you find a way around that, if you can keep your liver and your muscle clearing and making glucose, clearing it in the appropriate way, then you'll be fine. If, however, those tissues become resistant to insulin, as they often do, the so-called concept of insulin resistance, then it's over to your poor old pancreas, which has to work really hard and produce a lot of insulin. And the more insulin resistant you are, the more insulin it has to make. But of course, there are many people, probably many people in this room, who don't have diabetes, but have got a lot of insulin resistance, but their pancreas are fantastic. And we've seen people who are into their 80s and 90s without diabetes who have had to make phenomenal amounts of insulin all their lives, but they do it. Now, they may run into trouble with cardiovascular, other diseases, but they avoid diabetes by having a terrific pancreas. And a lot of that is down to the luck and inheritance of how you inherit how good your pancreatic reserve capability actually is. But I'm going to come back at the end. I'm going to finish with this slide at the end to really rehearse what we've learned as we've gone through the talk about these elements of the problem. Now, the first thing to say is you'd often think, listening to a lot of the press at the moment, that obesity is a new plague that's affected the world for the first time. Obesity is not a new disease. The Paleolithic sculpture of the Venus of Willendorf in 25,000 years BC, I don't think could have imagined this sort of, without having had any visual evidence that women of that shape did exist, and indeed were venerated in that era for their ability to acquire and maintain caloric positive energy balance. Hippocrates, that great Greek physician, was much troubled by how he should advise and treat his obese patients. He thought they should eat only once a day, take no baths, sleep on a hard bed, and walk naked as long as possible. Now, I don't think we've actually moved very far from that in our management over the last 2,000 years or more. And then obesity has always been around. The famous photo of Daniel Lambert, who ran the prison in Leicester in the 18th century, and a physician around that time opined, Thomas Short, no age has seen more instances of corpulency than our own. So we're not new, it's been around. But it has, of course, increased dramatically over the last 50 years, particularly 100 years. So it has been a condition which has had affected smaller numbers of people, and the distribution has shifted dramatically over the last 50 years. Now, I'm not an epidemiologist, I'm not a social scientist, but I do think that there is enough data and reasonable data out there to say that we can have some reasonable explanations for this recent increase in prevalence. Never in the history of human evolution have calories been as cheap, constantly presented to us, more attractive, and less filling because they are full of carbohydrate and fat rather than protein in many cases. Never in the whole history of humanity have we been exposed to these caloric load, this obesogenic environment. So no wonder, and that's true in almost most societies, even societies that, particularly developing societies now, there's been no time in human history where we've been able to get those calories without having to expend a vast amount of time digging, hunting, or physically expending calories to get them into our table. Because we have less physical activity in the workplace, in the domestic setting, less thermogenic demand because we have central heating, we have cooling, etc. So we've done everything to ensure that we can become obese because our food intake is almost infinite, our capacity to have food intake, and our need to expend energy has minimized. So it's not at all surprising that obesity has become common. People have suggested other factors, antibiotics, environmental pollutions, your antenatal environment, your mother makes you obese, or your friends, your social networks, your friends make you obese. But all of these are interesting suggestions, but I think that there's no real solid reproducible proof as yet that these are major impacts. And there are quirky genetic reasons why things might suddenly accelerate. There is such a thing as assortative mating, obese people marrying obese people more than they marry thin people, and then having a more rapid accumulation of genes. Or even in certain societies, obese women have far more children than non-obese women. So there's a differential fertility effect which can actually impact. But no doubt, I think that environmental factors are the most important. And I think probably the best data about the importance of environment is when people move from an area of low prevalence of obesity, such as rural China, and move to urban centers, either China or India. They tend to acquire the obesity prevalence of the place that they move to. So there is a huge, clearly effect, as illustrated by this particular demonstration here. So the real question is why, given this obesogenic environment, the question that interests me most is if it's also obesogenic and everyone is exposed to all this, then why aren't we all fat? Well, maybe we're all the same, and maybe that people who remain thin are just morally better than us who don't. They're just making conscious choices, they're clever, or they're lucky to live in environments that promotes leanness. That's, many lean people, I think, deep down probably feel that that's true. But it's perhaps more plausible to think that people have different susceptibilities to becoming lean or obese. This could be impervious to environmental factors, a bit unlikely, but could interact with environmental factors. And I think what I'm going to spend the next period of time doing is to try and convince you that these statements are the true ones, that people do have greatly different susceptibilities to becoming lean or obese based on their intrinsic genetics. But that interacts very strongly with environmental factors, and the two are not at all mutually exclusive. So we know that genetics is important because we have got wonderful and very, very rich data from studies of identical and non-identical twins, from people who've been adopted, or families that they've been adopted into, from identical twins who have even been reared apart. So we have a remarkable data. And there's very strong evidence for a very high heritability. It's not quite as heritable as height, but it is very, very heritable phenotype, your body mass index. And what I mean by heritability is it means that if I take this audience and take two random people and imagine I took out all their fat and put it in a weighing scales and the other person, and there's a difference between those two people. I ask, of that difference between these two people's fat mass, what percentage of that is down to genetics? And you'll be surprised to hear that even in the modern obesity era, the general accepted figure is somewhere between 50 and 70% of that is down to genetic differences that they share between the two groups. And the most compelling observation ever made, and it won't be done again, I hope, because this sort of practice doesn't happen but it did happen in the 1950s in Sweden, where there was a lot of adoption in Sweden. There was a lot of premarital sex. There was very little contraception. There were loads of people born out of wedlock. Swedes were quite wealthy, but they wanted to adopt one child, not two children. A lot of twins brought up for adoption in Sweden, but the Swedish authorities decided that they would separate those twins at birth and allow them to be brought up by different families, and they never knew that they had an adopted twin. And so that was the policy for many decades in Sweden. That gave an opportunity to do a remarkable observation, to say, what's more important for your adult weight? The family that brought you up, or the twin that you've never seen? And the remarkable work done by Mickey Stunkard, a child psychiatrist from Philadelphia, who went to Sweden to look at this data and find the data, and he scratched his head when he saw the results, because it was not what he was expecting. He actually had to conclude from his data, as you can see, the childhood environment has little or no influence. There was no correlation between the adult weight of the people and the family that brought them up, their brothers and sisters that they grew up with. There was zero. Correlation was actually zero. It wasn't just low, it was random. And the correlation with the identical twin that they never saw was 0.75. In other words, they would really lay on the line, a very, very tight line, and these people had never seen each other. So this experiment has never been repeated, obviously, but it's very compelling and highly informative, I believe. And for those people who prefer pictures, this is not from that particular study, but these are just a set of photographs taken from sisters born on the same day, but sharing 50% of their genes because they're non-identical twins. And you can see, like you and your brothers and sisters, you come in all shapes and sizes because half of your genes are different. And so they come in all shapes and sizes. Here are sisters born on the same day, sharing 100% of their genes. Difficult to look at that picture and say, well, I don't think genes are very important. I mean, it's pretty remarkable in terms of both the amount and the distribution of body fat that genetics is extraordinarily important in determining the shape we are as we age. So I often say at this stage of the point, I'm going to put this up and then I'm going to go away because that's the answer to everything. This is the end of the talk. The cause of obesity is extraordinarily simple. It's energy stored equals energy intake minus energy expended. And I mean, it's simply thermodynamics, and that is the truth. As a thermodynamic organ, as a piece of the world, that is true. We cannot add energy to ourselves without violating that law. We can, of course, store the energy in different ways. We could store it as fat or as non-fat tissues. I've often dreamed I could go out for a slap-up curry, and instead of putting on weight, I put it into a vigorous beard growth overnight that I could cut off in the morning. But that would be a wonderful way to partition energy in a different way. But unfortunately, we can't do that. So nutrient partitioning is a way that you can have differential putting it into fat and non-fat tissues. We do get through a remarkable amount of food in our lifetime. And it's remarkable that we're not all oscillating between Kate Moss and Luciano Pavarotti throughout our lives. We tend to stay on similar centiles, even though we're not probably consciously saying, oh, I want to eat the same every today or tomorrow. But by and large, we do remain pretty stable. And that stability struck some scientists at the beginning of the last century and wondered, what governs that stability, relative stability of body weight? If you take a rat and keep it in the laboratory, rats keep growing forever, by the way. So if you see a big rat, it's an old rat. So they don't fuse their growth plates, so they just keep growing throughout life. And what Kennedy and others, Gordon Kennedy, a British physiologist, and other workers in the States did some very simple experiments. They just restricted rats' food to 50% rations for 2 weeks. And of course, it lost weight and didn't keep going. But then they put it back, and they said, well, what will it do when you put it back under normal rations? Well, very rapidly, it ate about double what it needed and went straight back to the trajectory it was on before and stayed on that trajectory, exactly like the rat that had normal food. And the same is true if you overfeed them with gavage feeding and too much food. So the rat knows what weight it wants to be, and how does it know, and what knows, and where does it know? Which bit of the body knows this? And we've had a clue for hundreds of years from patients who may have normal body weight, and then in middle life suddenly become catastrophically obese and get a very bad headache, and sometimes have an epileptic fit, and in the old days died, and at post-mortem were found to have tumors in the hypothalamus of the brain, an area of the brain which we now know to be crucial for the control of body weight. A tiny area just in a few inches in the back of your nose, deep part of the brain, this tiny area. And we know it's important because that led to scientists doing much more selective, rather than waiting for tumors to develop, taking little electrolytic lesions and making burn a small area of the hypothalamus. And if you do that in the middle of the hypothalamus, in the so-called the ventromedial hypothalamus, you create a vastly overeating and huge rat that will never stop eating. But if you move the cannulae just a few nanometers to the left or millimeters to the left or right, you can create a rat that will never eat again. It will drink, it will copulate, it will do everything else, but it will starve itself to death and never feel hungry. And that is just a sub-millimeter area away from the bit of the hypothalamus that controls food intake and makes you severely obese. So this experiment, which we wouldn't be able to do these days, it's kind of beyond British Home Office what they would permit, but nonetheless, it was crucial for the modern era. And it was a so-called parabiosis experiment, where you take neonatal rats, take newborn rats, and you partly put their circulations together by sewing the skin together of the two rats. And this was done by a chap called Harvey, who I just discovered a few months ago is still alive at 95 in Leeds. And he worked with David McCance, who was the first professor of experimental medicine here. And this was McCance's idea. McCance was a great experimentalist and physician scientist, and the first MRC professor of experimental medicine here. And during the war, Widdowson developed all the food tables that kept Britain alive during the Second World War. But he was a brilliant experimentalist, and he suggested to a young PhD student, Harvey, that these experiments that the guys in the States had done about controlling body weight, there might be a circulating factor. And the way of finding out if there was a circulating factor controlling weight was to put these animals together. So Harvey did this experiment, lesioned, tried to make this guy very obese. And what happened was this guy got very obese, but this guy got extremely thin, lost interest in eating, and suppressed food intake and became very lean. And this was the first evidence that a circulating substance was present in a very obese animal that was suppressing food intake in the lean animal. And really, I think is the beginning of the modern era of thinking about the physiology of body weight and the control of obesity. This was done, by the way, a few hundred yards from here. You know, the Judge Institute now, it was the old Addenbrooke's Hospital, and that had a small laboratory in it, and that was done in 1958 or so, just a few hundred yards from here. So Doug Coleman, who worked in the Jackson Labs in Bar Harbor, Maine, and the Jackson Labs is an amazing place which has thousands, started off in the beginning of the 1900s as a mouse menagerie. All the mouse fanciers who kept different colored mice and loved keeping pet mice, when they got a particular set that they loved the color of, they wanted to keep them breeding forever. So the Jackson Labs agreed to keep a breeding pair of all these lovely different color and furry mice, and thinking that in the long term it might be useful for medical research. And it was, because some of the mice suddenly that appeared in their strain suddenly became hugely obese spontaneously, the so-called obese mice, the OB mouse. And that bred true. Mice who had two copies of the OB gene were enormously fat. And so what Coleman did was he did the same experiment as Harvey. He did the parabiosis, together, took a normal mouse and stitched it up to the OB mouse. And lo and behold, it completely cured the obesity of the OB. So a normal mouse, something was present circulating that was lacking in the OB mouse and caused it to, when put together, caused it to be normalized. And that, of course, led in the 1990s to the discovery by Jeff Friedman at Rockefeller Institute of what was missing from this guy compared to his two siblings. And what was missing was a new hormone. And that new hormone he called leptin, from the Greek for thin, leptos, and it was only made by the fat cell. So literally the fat cell was, at that stage, thought to be a very boring, tedious, passive receptacle of our fat. Suddenly it turned within minutes, at the appearance of this paper, into an intelligent member of the neuroendocrine signaling community, talking through its hormones to brain. And so the OB animals who lacked leptin coming from the fat cell, if you just gave them a tiny amount of leptin directly into the hypothalamus in the brain, everything got better. Totally cured every aspect of obesity in the OB animal. And this discovery has really, again, catalyzed the very modern era of obesity research over the last 20 years or more, and it has caused us and the whole field to move from a backwater into a really hot area of biomedical research. And so what's happened over the last 20 years are numerous discoveries. Leptin is a long-term signal that goes from our fat to our brain, keeping our brain informed about the state of long-term fat stores and how much fat we're keeping. But we've learned over the last decade or more now about short-term hormone signals. So when we eat, a whole range of hormones come from little endocrine cells in our gut, some of whom are shown here. And they modulate our short-term food intake. So the reason we feel very full after a large steak and less so after the same amount of calories but given as, let's say, chocolate chip ice cream, is that the large steak induces an enormous release of these hormones. It's very protein sensitive, and so they suppress food intake very powerfully after the ingestion of protein-rich meals, particularly. But they act in the short term rather than the long term. And we've added to those recently, work we've done with a local company, but also Fiona Gribble Institute. I've added another hormone recently to that panoply of gut-derived peptides. And we're really only beginning to learn how they're integrated and how they all work together. It's a very fascinating and subtle interaction. That work of the gut hormones has actually led to the only licensed therapy for obesity in Europe and America, not yet licensed in this country for the treatment of obesity, but one of the hormones modified, one of those gut hormones, is called liraglutide. And you can see that it can reduce in about a 10% body weight loss. That can be up to, in the more obese people, up to 15 kilos of weight loss with longer term treatment. So it's not totally effective, but it's not a bad anti-obesity treatment, and by and large safe, because it's based on a normal hormone coming from the gut. So I think we are at a new era. Many companies are trying to mix these hormones and find the right mixture that together might give you a bigger satiety signal than the ones than using a single hormone alone. So all of this science on the mice, we are very fortunate. This is where I said wandering into by accident, but my own adventure in obesity came from studying this patient in my own clinic. I was seeing her for other reasons other than her obesity, but we eventually turned out and discovered why she was obese and what genetic cause that was in the brain, an enzyme that was not working in her brain that processed many of these leptin signals to downstream signals. And shortly after that, because we got very interested in obesity, we found the first children who actually were like the OB mouse. They too lacked leptin. So leptin was in fact not just a mouse thing, it's a human thing. There are children around who are severely obese, constantly seeking food, obese from a young age, immune to any advice or education or impact because they are totally driven, eat effectively torpedo eating machines. They're constantly focused on finding food, no matter what the parents do. And that's because they lack this signal leptin, which in effect means they feel that they're starving all the time. And that launched an era for me personally and for the field with wonderful colleagues that joined me around the time, Sadaf Faruqi, Giles Yeo, and Tony Call, all of whom have remained in the institute and have been now running their own laboratories. And we discovered many genes that, when disrupted, caused obesity in humans. Not just the first two that we found, but many genes. And when we found these genes, and asked where they're expressed in the organism, in the human and the mouse, these turned out to all be brain genes, and very much in this part of the hypothalamus is where they were doing their work. And when we actually take the children who carried these mutations into the clinical research facility and ask, why are they obese? Why have they become obese when they have these broken genes? And we measure how much energy they expend, and we measure how much food they take in. If we give them an ad libitum meal, we find very profoundly that they just eat a vast amount. They do not have normal satiety. They do not have normal. Here's a leptin deficient children eating 3 times as much as control children, corrected even for their body mass. So vastly hyperphagic, as we call it. And even with these condition MC4R, we could take the mutation, put it into a test tube, measure how dysfunctional it was in the test tube, and that could predict how much food these people ate when they these children ate when they came into the. So here was a single molecule in the brain, and how it was working was actually very strongly influencing how much people would want to eat at a single sitting. So it's a profound impact of a molecule on eating behavior. And the work in childhood obesity is now handed over by me to the leadership of my colleague Sadaf Faruqi and Inez Barroso at the Sanger Center, who's a great geneticist, and more such defects have been found, and more recently by other groups continuing. So this discovery has not stopped. But it's reasonable to ask, these are all rare, relatively rare conditions. I mean, the MC4R is not that rare. About 1 in 1,000 people in Britain, about 50,000 people will be obese in Britain because they carry a mutant form of melanocortin-4 receptor. That's commoner than cystic fibrosis or Duchenne muscular dystrophy or any of these conditions you know about. But what about common obesity, common BMI? What about the range in this room from the slimmest person to the most obese person in this room? Are these processes and genes of any relevance to those? Well, we kind of know that. This slide is slightly out of date. We now have data from over 1 million people worldwide who have been genetic fingerprinted at most common and even low frequency genetic variation across the population have been studied in 1 million people. So this data is unbelievably statistically robust. The p-values are completely up the wazoo. There's no doubt about the robustness of the associations of these data. So you can look at this stage when it was only 300,000 people. There were 97 places in your genome which, if you had a particular variant there, you were either more or less susceptible to being obese, and that was highly significant. Now you could take those 97 and say, what do they look like? Where are those genes? Where are they expressed? What bit of the body? Are they all in fat cells? Are they all sucking in fat? Do you become fat when you have these aggressive fat cells that are picking up fat from the circulation and putting it in to the fat cell? Well, the answer is no. The answer is these genes are pretty much all operating in the central nervous system. So again, like our severely obese children, which is effectively have a neurobehavioral disease, in this room, your relative susceptibility to obesity is determined by genes that are operating in your brain. and not very much in your periphery or your fat or elsewhere. And this was kind of nicely put together by Jane Wardle, a wonderful colleague who died very tragically, far too young, a couple of years ago. And Jane was a great child psychologist, and she had worked for many years developing questionnaires to work out what sort of behaviors in children would lead them to become obese as they got older. And she developed this food responsiveness and satiety responsiveness questionnaires, which were good at predicting what kids would go on to develop obesity and what sort of behavioral traits. And what she and we were able to do was able to take those genetic variants and say, do they influence these traits that Jane had discovered and described? And it was beautiful in that, yes, those genetic variants, we saw their genes are expressed in the brain. They do track along these satiety responsiveness traits, and that does predict your body mass index. So here we had a link between the gene in the brain and behavioral traits in children that then translated into body mass index. And this is kind of my final slide on the causes of obesity, but it's very simple and it's adapted from Jane, but it's a good take-home message, but it's extremely simple. You know, a lot of more thuggish medical colleagues of mine will be very brutal about people with obesity and say, well, nobody fat ever came out of a camp where they were starved. It's just, well, of course they didn't, because you need energy in to. So of course, in conditions of famine or forced famine, you will never get obesity. But that's almost beside the point. If we all lived on no oxygen, it wouldn't matter whether we had bad lungs or good lungs. We'd have none of us would have any oxygen. So Um, but in famine, nobody's obese, of course. But as soon as we get round to having a food supply, then there are some people who are going to become obese and some people not. And of course, now we have an abundant or even superabundant food supply. Even those of medium susceptibilities are likely to become obese. And I guess what our work and others have done is to show that those genetic susceptibilities are largely around food intake. There may be some genes that influence whether we like to jog or not, or whether we like to twitch or not, or whether we like to burn off calories or not. But from what we've seen so far, the majority of the genetic influences that we can find are genetic influences on food and food-related behavior, which does, I think, have a public health message. It means if we're going to change something, to change the environment to fit our biology, we need to be presenting less food and less frequent food and less. We need to create an environment which is less easy to immediately for those individuals who are very susceptible to eat in the presence of a stimulus. Has the work we've done any good? Well, we've been very fortunate in that one of the conditions we found, the leptin deficiency, is beautifully treatable with the injection of leptin. So we can take the children who are severely obese with leptin deficiency, and in every case, this is a very rare condition. We know only about 70 children worldwide. We're treating about 30 of those. Sadly, many of those at the moment that we know about are in countries that are very difficult to access and get help to. But it's remarkable because many of these children are effectively wheelchair-bound with the severity of their obesity. And within months now, we can essentially turn them back into completely normal children. So this is a great proof that leptin truly is a human hormone. Without it, we turn into an organism that just seeks food all the time and becomes vastly obese. And with leptin, we totally reverse the defect. I think one of the nicest experiments we've ever done is with our colleague Paul Fletcher, a neuroimager and psychiatrist. And these are studies of two young adolescents who had no leptin and were undiagnosed. They came from Egypt and Turkey. And they agreed to be studied in the scanner, the MRI scanner, before and after we gave them leptin. So when we put them in the scanner before leptin and showed them pictures of footballers, not much happened. Showed them pictures of money, not much happened. Showed them pictures of cars, not much. Showed them pictures of food, and the areas of addiction and reward turned on like you'd shown a drug addict the paraphernalia of cocaine addiction. Massive increase in blood flow in the areas. And that was even broccoli. So any food stuff. And then within 3 days of treating with leptin, and within 3 days they have not changed their shape or weight or they have not changed anything. But within 3 days, these are the differences here, pre and post leptin. 3 days, we had completely normalized that response. There was no, they were just like normal individuals. So here's leptin, a little hormone coming from your fat cell. And right now in every one of you, it's going to your brain and it's telling your brain, it's okay, you're not starving. You don't have to immediately leave this room and go and get food because you've got enough nutrition on board. So every one of you is having that signal right now, because if you didn't have it, you'd be exactly like this child here, and you wouldn't be able to concentrate and listen to my talk because the only thought in your mind would be that you need to go and find food. So leptin is working in all of you right now. Why isn't it a blockbuster drug then? It works incredibly well in this situation where you have very marked obesity, and then after leptin we can restore you to normal. It works remarkably well in another condition. This is a condition where you can't make fat cells, so-called lipodystrophy. And that's a terrible condition because A, you don't have fat cells, and I told you how important they are, and B, the fat cells make leptin, so you don't have leptin. So these unfortunate people are starving, but then when they eat, they've got nowhere to put that extra fat. And you can see in this case, I don't know if you can see it, those spots are actually lipid spots. It's fat coming out the skin. These people are disastrously metabolically ill. But when you give them, and this is the liver is enormously swollen. And here's leptin treatment in this condition, which reduces the appetite, even though they don't have any fat. At least they're not eating and pouring fat into the rest of their organs. But unfortunately, when you give leptin to regular obese people, this is one of the original studies which depressed the pharmaceutical industry, who spent many millions on buying the patent. it wasn't terribly powerful. Some people lost quite a bit of weight, but a lot of people didn't lose much weight, and leptin wasn't terribly effective in reducing body weight in regular obesity. And that's because leptin doesn't work like this. Many hormones, if you give more of it, you get more of an effect. But leptin looks more like this. If you give somebody with no leptin some, you have profound effects, but very quickly it flattens off. The shape of the dose-response curve is different. So it's either it's certainly not model 2, but we could have a model of 3. So what is important to recognize is that leptin probably existed not as an obesity-preventing hormone, but it existed to signal starvation. Its real job is like Sherlock Holmes' dog that didn't bark in the night. It does most of its signaling in its absence rather than its presence. It's the absence of leptin that screams at the brain, oh my God, you're starving. And once it's present, you kind of ignore it, and because it's there enough. So giving more under those circumstances probably is the reason it doesn't seem to do very much more. Having said that, we do have hints from people. Here are the parents of kids with leptin deficiency who carry one mutant copy of the leptin gene, and you can see they tend to be a bit heavier, and they tend to have lower leptins, the ones in blue. So even a subtle lack of leptin can give you a little tendency to be obese. And if you take 12,000 people in East Anglia, some of you may be in this study. This is the Fenland study, 12,000. I'm sure if you come to this meeting, there's probably a disproportionate number of you are in the Fenland study. And here is the log of leptin and obese. Here's individuals who are obese. So in Fenland, here's 4 individuals whose leptin levels are incredibly low for their degree of obesity. So we're very excited about trying to find such individuals and trying to see if they are a subset of obese people who might actually respond to leptin supplementation. And that's work that's ongoing at the moment. In small numbers, as you can see, that's 12,000, and here we have 5. So these are not going to be a blockbuster drug. But on the other hand, for those individuals, if they truly are partially leptin deficient, it could be transformative. And there are other new drugs based on downstream of leptin. Here's the pro-piomelanocortin, or POMC, is a signal that's the next relay station along in the brain. And people who lack that are severely obese. And here is their weight when you give back the active portion of POMC to those individuals. And we're working with this compound in other patients now with that condition. A little amusing aside, I mentioned that we share a lot of biology with other species that we share the planet with, and we're certainly not alone in our modern problem with obesity. Canine obesity is also an epidemic, and one that Labradors seem to be particularly prone to. And Eleanor Raffin, working in the lab as a vet, convinced me that we should invest a little bit in seeing if we could learn something from our friend, the dog. And very recently, and kind of almost serendipitously, we discovered that the Labrador carries quite a large proportion of Labradors carry a deletion of one of these genes that's downstream of leptin. And they're the Labradors that have got increased food motivation, a greater tendency to become obese, and they might even have a greater trainability. They're more highly prevalent among guide dogs, because the idea being maybe you can train them easier because they're more motivated for the training that we can actually give them. So I'm going to move to the last part of the talk, which is really to tell you not about what causes obesity, but what the consequences of obesity are. Why is it so bad to be fat? Why does having an energy intake more than your expenditure do you bad? I don't really like the term obesity. I think it leads us to brain freeze. I think some people hear it and then they stop thinking. They think they know what it is, but all it means is an expanded mass of triacylglycerol, triglyceride, in your adipose tissue, and with a relatively arbitrary cutoff between what's normal and what's overweight and what's obese. But there certainly are associations, and with adverse effects, and I've divided them into 3 types. Mechanical and gravitational, the sort of things that simply being heavy result in too much weight on your knees, certainly contribute to osteoarthritis. Too much weight in your abdomen pressing on your esophagus probably contributes to reflux esophagitis. Too much fat in your soft palate contributes to narrowing your airway and giving you sleep apnea. They're kind of mechanical adverse effects of obesity. But it's not immediately obvious why obesity should lead to certain cancers or certain metabolic diseases, but they are strongly associated. Is it, though, however, that it's the obesity that's causing these? Or, in fact, is it that the obesity is just an innocent bystander, and it's something about your energy intake being more than your energy expenditure that's causing these nasty, abnormal conditions, particularly metabolic disease? I'm not going to talk about cancer today. It's not an area of my own particular expertise, but metabolic disease is what I've been studying for many years, so I'll talk briefly about that. And the first I put in there, I mentioned to you at the very beginning that one of the earliest things that happens when fat goes in the wrong place is that you become resistant to the actions of insulin on carbohydrate metabolism. And insulin resistance is one of the earliest abnormalities we see. So it varies enormously. As people get fatter, some people become very insulin resistant. These are all non-diabetic people, but they have very, very different levels of insulin required to keep them non-diabetic. Some people get fat but don't get insulin resistant. Some people get fat and get very insulin resistant, but that's that sort of relationship. And it's understanding what makes some people like that and some people like that is one of the main interests in our laboratory. This is data from my co-director of our institute and director of the MRC epidemiology unit, Nick Wareham. So how do we get from obesity to insulin resistance? If you were to ask 100 diabetes specialists now who read the literature and read what they made in the journals and conference proceedings, they would say, well, we think we know now. We know because we've discovered that when you get an obese fat mass, it becomes inflamed. And it's these inflammatory cells that go into fat, and they release a lot of these cytokines. And it's those guys that are the bad guys, going to liver and muscle and causing a disruption of glucose clearance. I've got a slightly older idea, but one that attracts me more. And that is that it's sort of, you can put it that like that your big fat cell is not your enemy. Your fat cell is your friend. And it's only when your fat cell can no longer do its job of storing that fat and you start having to redirect fat into other tissues, then you get that ectopic fat moving into liver and muscle. So it's doing its damnedest to try and keep you safe, and your fat cell is working for you, but it's got a limited capacity. And when it stops being able to control enough fat, that's your total fat mass, then it's when you get the ectopic fat going and causing the mischief in liver and muscle. And I'm inclined towards that because of our studies and experience in people with this condition, lipodystrophy, that I mentioned. These are people who can't make enough fat, either no fat at all because they literally can't make fat cells, or they can't make fat cells in particular parts of their body. They have a limited ability to make fat cells. They have genetic conditions. And our lab, particularly David Savage, working with me for years and now a PI, we've discovered many of the genes that cause lipodystrophy. And I'm going to give you an example of one of those genes in a moment, because I think it's very illuminating. Here's a fat cell. Here's what if you take an electron microscope of fat tissue, it looks a bit like that, each of the fat cells being a sphere, in effect. And the reason why fat cells were thought to be hilariously boring, because they're 99% one thing. 99% of the volume is a big blob of triacylglycerol or triglyceride, surrounded by a monolayer of phospholipid around, of polar lipid just around that, and was described at one stage as looking like a fried egg on a beach ball. So the fried egg being the nucleus and the cytoplasm, and the beach ball being the fat droplet. So each of your fat cells looks like that, and it looks incredibly boring, and it's all the same. But of course, it's a beautifully regulated organelle and a perfect design to do its job. And one condition that we described a few years ago affects a molecule that's only working on the surface of that droplet. That's its only job, is to be a guardian of that fat droplet. And every person in the world who has a mutation of a particular type in that, all of them develop this condition, and if they inherited it from their mother or father, they all get it, and basically you can't tolerate messing around even with one copy of this. You get this condition. Here they all get insulin resistance, diabetes. You see, they don't look like typical diabetic people. They actually look slim. It's because they can't make enough fat cells. Their fat cells just can't hold on to the lipid. And I'm going to please bear with me for a little bit of biochemistry. It's kind of cartoon biochemistry, but I think we don't really understand things unless we understand them at the atomic level, in my view. And I think it's important to really go a little deep. So here's what the fat cell droplet looks like. And when you are feeding these guys, this perilipin and its pal, CGI58, they sit on the surface. And these are the enzymes that break down fat, ATGL and HSL. But of course, when you're feeding, you don't want to break down fat. You're getting fat in your diet. So you keep them away from the surface of the droplet. And that's what happens. You just make and put triglyceride into droplets. But tonight, when you go to sleep, and about 4 or 5 hours after your last meal, this process will happen to all of you as you go to sleep about the middle of the night. And this will happen to every one of you tonight. And without it, you'll all be dead. So this is a really important process. And what happens is that because you change your hormonal milieu overnight, you start getting what are called biochemical changes in the fat cell that put phosphate groups onto perilipin. And one of the things that happens is that the C-terminus of perilipin is phosphorylated. That kicks this guy off, and he goes whizzing around the surface of the droplet looking for the first enzyme to break down triglyceride, the ATGL, to start breaking the triglyceride down to diglyceride and free fatty acid. And then beautifully coordinated, the N-terminal phosphorylation event grabs the second guy, and this second guy, HSL, breaks down the diglyceride to free fatty acids. So overnight, when your heart is beating, your heart needs free fatty acids. Your brain needs glucose, but while your brain is busy taking up all the glucose you're making, other tissues, particularly your heart, need sources of fuel. And that source of fuel is largely the free fatty acids that you come from your adipose tissue overnight. And the reason you can do that is you have this beautiful system coordinated by these two guardians, perilipin and CGI58. Now, what is happening to all those poor people whose picture I showed you in the previous thing, these people, what is happening to them is that all the time they have one duff copy of perilipin with a messed up end to it that can't get this signal. So all the time it can't partner with this guy. So even when they're being fed, this signal is going around grabbing the fat breakdown enzyme and making it break down fat in fat cells. So even when you're eating, your fat cells are releasing fatty acids, and they are going to other tissues and mucking them up by going into the muscle and liver. And that's what's causing the insulin resistance, fatty liver, and atherosclerosis. And the thing we got most excited about in the last year is to find that this just isn't a rarity. This is actually the commonest cause of metabolic syndrome in the world, because a little subtle variance of this failure of fat storage and failure of breakdown is probably at the heart of much of metabolic disease across the globe. We did this by working with our epidemiological colleagues, and they got access to remarkable amounts of data now. Nearly 250,000 people now, genetic variation of 2.5 million variants across the genome. We can ask very specific questions of them. We can say, what are the genetic variants in the population that make us be insulin resistant with high blood fats and low blood HDL? And they found that there are 53 of those variants in the population. And when you look at those, here are the list of the genes and the SNPs that they're beside, and the red is increased, the blue is decreased, the red is increased. This is how we defined the phenotype, so of course they'd be that color. But here are those 53 genes, and here you can see reds for coronary artery disease and types. So these are gene variants that predispose to coronary artery disease in type 2. But the remarkable bit is the blue bit here. In other words, these are genetic risk factors. If you have, at any one gene, you can have one bad copy, two bad copies, or no bad copy. If you take the bad copies of those and add them up, you go then to a general population and say, are those genes associated with obesity? They're actually associated with thinness. These are genes that are associated with low body fat. And you can see here, highly significantly associated with lower body fat percentage. And particularly, they're associated with having low amounts of fat on your bum and on your legs. So these genetic variants that predispose in the general population to you developing insulin resistance and metabolic syndrome are actually genes that make it hard for you to put safe fat on your bottom or on your legs. You can see that android fat, there's an increase. If you have got genes that make you centrally obese, they do push you in the different direction. But much more importantly is your inability to put fat on your legs. These are adipose genes. This is the same sort of bioinformatic process that we looked at. Which tissues are they important in? They're important in adipose tissue. And then when we play around them with fat cells, we can show that they are truly important for the differentiation of fat cells. Now, I'm going to turn this more into a lay speak in a moment, because I think this bathroom, the soggy bathroom carpet analogy, is probably my favorite analogy for this and what we've found. Here is your bath, and here's the amount of water in your bath. And that's the amount of potential safe. The bath is how much you can potentially store as safe fat, and that's how much you actually have, is where the bath is. And where you get to when you turn the tap on, leave it on, and you forget to put the plug in and leave it out, is you reach a steady state. And if your steady state is there, then you're laughing. Then you've got a safe amount of fat stored. You're not getting a soggy bathroom carpet. It's all nice and clean, and nothing is going wrong. Now, traditionally, when we think about obesity, and the reason obesity leads to insulin resistance and diabetes, is that we have too much energy coming in, or not enough energy coming out, or certainly an imbalance between the in and the out. And then, of course, it's easy to overfill your bath. And once you overfill the bath, then you spill out and cause a horrible mess in the carpet. And that's when you get the mess that we call diabetes, where it spills out into tissues that it shouldn't be. And that's kind of, people haven't pictured it like that, but that's how the world has sort of thought about it. And it's a very simple way of thinking about it. I think what our research in lipodystrophy and in low body fat shows is that, in fact, what's really important is the size of your bath. Because many people become soggy carpeted at much lower levels of adipose storage, because they simply don't have the ability to safely expand their adipose mass. So individuals become insulin resistant at relatively low amounts of energy imbalance. And this may indeed be why, for example, in South Asia, most diabetes occurs in individuals we would not consider to be obese. By the time they're even somewhat overweight, they have already developed insulin resistance and diabetes, presumably because they do not have these enormous baths. And in fact, the phenomenon of the metabolically normal obese, the enormously obese person who is not diabetic, is very strongly a Western phenomenon. That is a phenotype that we see more often in Western people than almost anywhere else. In fact, if you ever read a newspaper and someone has had to have the ambulance service or the fire brigade come and knock down the door to get them to hospital because they've been so huge as to be stuck in their house, they almost never have diabetes. And the reason that is that they have an enormous bath. So they are the people who have been able to get bigger and bigger and bigger, and they've never decompensated to get diabetes. Because as soon as you get diabetes, of course, you lose weight, because you pee out the calories as glucose in your urine. So only the people who don't develop this overspill are the ones who get massively, massively obese. So are there any practical implications of this? Well, we run a national service for people with severe forms of insulin resistance, many of whom have difficulty putting fat on their limbs. This is a lady, you can see she's got plenty of central fat, but really for a woman, very little fat on the limbs, and this is a DEXA scan. So she has a classic partial lipodystrophy, an inability to put fat in these safe fat places. She has terrible diabetes, terrible control, many, many hundreds of units of insulin, been sick for years, has complications, and we do something, and by April the 23rd, 2013, she's pretty normal. She's completely normalized, she's off all drugs, she is completely effectively cured. And why she's cured is that we haven't actually changed the size of her bath, because we don't really have ways of doing that. But what we've done is we've turned the tap off. And the way we've turned the tap off is by operating on her gastrointestinal tract so that she gets all the signals to stop her eating. And so she has a profound reduction in food intake, because we have given her, in effect, an operation that changes her hormonal milieu and makes her satisfied with very small meals. So within 3 or 4 months of that, she is profoundly altered by her bariatric surgery. So now we can see how sensitive these people are with small fat storage depots to be able to change. They are particularly benefit from procedures that reduce food intake. So to come to the end, what have we learned over the talk? Well, we've learned that our brain is really important. Genetics tells us that brain's really important. What we've learned about the poor old boring fat cell is it's crucial its health is really central to a lot of metabolic health. It's not just a boring recipient. It's not just cosmetically tedious. It's not just there to cause cellulite. It's a really important tissue to maintain health. And I haven't talked so much about liver and muscle because we haven't learned so much about those. I also haven't talked about pancreas because that's largely work of others in the UK and elsewhere about who has and who has not got good pancreases. But that will determine largely whether you get the high blood glucose or not. And then at last, I think in a public talk, I have given you an unashamedly biomedical and biological approach. I think it's reasonable for you to give a last few slides on asking whether this has any relevance to tackling obesity as a public health problem. Is obesity, for example, like smoking? We've done pretty well on smoking, and we can have a successful influence on both individual and population behavior through providing information, facilitating non-smoking, incentivizing it, even coercing it. And these various ways have been extremely effective at reducing smoking. Of course, smoking is one thing, because it's not a normal thing to do, if you like, but much rather different at eating and drinking, because every one of us, of course, has to consume some calories, and banning eating or banning drinking is just not going to be feasible. So it's obviously more challenging. Do we have a useful, if smoking isn't a useful precedent, are there any other useful precedents? Well, I think that hypertension or high blood pressure is a reasonable model. For example, hypertension is just the higher end of a blood pressure distribution. It's just being over there, an arbitrary cutoff between normal and high, but really it's a continuum. We just stick a thing there. And obesity, similarly, is the higher end of a distribution of amount of body fat or adiposity that we get. So they're really very similar. It is true that if you're at that end of either blood pressure or that end of weight, you get nasty consequences. It's not that it's not, I'm not saying it's trivial, but it is nonetheless a continuum. So they're both continuums of distributions. So where are we with hypertension? Well, even 50 years ago, and even when I was training some 30 years ago, the wards were full of people with uncontrolled hypertension, having strokes, having renal failure, having heart failure. Hypertension was a major clinical problem in British hospitals 30, 40 years ago. Lots of our patients, people come in, so-called malignant hypertension. It was a huge problem. It's effectively disappeared as a hospitalization problem now, and we don't see anything like what we used to see in terms of hypertension. So there's a problem that effectively we've done something about pretty well. And how have we done it? Well, it's taken a long time, because the first approaches to people with very bad high blood pressure were these terrible rice-only diets with no salt. People found them intolerable, and they really couldn't stick with them for more than a few weeks, because they were so disgusting. And then in Boston in the 1940s, people with very severe high blood pressure were given surgical sympathectomies. They actually had operations on their neck to control the sympathetic nerve. That worked very well, but many of them could never stand up again because their blood pressures would drop to their boots when they stood up. So very invasive and very... Then in the 1950s, some drugs came in. Their drugs were effective, but they had awful side effects. And it made people depressed and suicidal and terrible, terrible drugs. And then gradually, we started having drugs that were a little bit better, and we started understanding a bit more about the control of blood pressure and how important the kidney was and how the kidney handled its salt and nutrients. We started doing science on the kidney and understanding how it worked. And now what we have are a whole series of agents that are now cost pennies, and that together there's almost nobody whose blood pressure cannot be controlled with a combination of these agents. Now, as well as that, we've had population approaches to reduce salt intake and to reduce salt that's routinely put into food. So a combination of a kind of classic biomedical science approach with a public health approach has been very effective in hypertension. But of course, where are we? We're kind of, if you look back at where we are now in obesity compared to what's happened with hypertension, we know that diet and exercise does work temporarily in most people, but it's just very difficult for obese people to sustain that long term, and the drive for their weight to go back is very profound. So much as we'd love to say all you need to do is diet and exercise, that's easy to say but very difficult to implement, and multiple studies have shown how difficult it is for people to maintain that over long periods of time. Some people do, they're remarkable and should be celebrated. It's not impossible, but for the vast majority of people, it is exceptionally difficult to get from a state, particularly from a severely obese state. And bariatric surgery is remarkably effective and indeed even cost effective, but it is pretty brutal in its way. Even though it's laparoscopic, it's a scary thing to think, and probably won't be widely usable for all. I think we use it inadequately in this country, but it certainly isn't an answer to everything. And then, of course, we're in the same stage now with obesity drug development as we were in the 1950s with hypertension drug development. We keep getting the stuff that really works well, makes you sick, and the stuff that is safe doesn't work terribly well. And so we've been in a bit of a bind with those, but slow progress being made. So I think that we need a multidisciplinary approach. There's been, for example, in the hypertension, we've mixed a policy that's designed to reduce salt intake and screening. These are epidemiological, population-based things. And now we have drugs which are generic, cheap, and can be combined. And I think that if we're going to do this same with obesity, we will need that combination. We need that combination of selective, specific health measures focused on prevention and the selective use of cheap, effective, and safe medications. Now, I mentioned that I'm not a population health person, or I'm not a public health person, but I will show one slide that I think might counter some. It's very easy for us all to have an idea of what we think might work to solve the obesity problem. And if you ask people, and generally lay audiences, most people think that it will be solved by education, and particularly educating in the young. And here's a paper that's just been published in the BMJ, one of the best studies of such most intensive and most rigorous ever undertaken. And this took several hundred schools in the Midlands and randomized the schools, to primary schools, to some schools that had a very intensive program, including 30 minutes extra per day for physical exercise, family-based education about food preparation, 6 weeks visits to the Aston Villa football over a 6-week period to get motivational talks from professional footballers, an immense amount of time spent on the children, and then the other schools had no such intervention. And at the end of this, here's the commentary this is the people's own commentary, is that it did not result in any meaningful effect on adiposity, dietary intake, or physical activity after 15 or 30 months. So an enormous amount of effort put into those schools with multidisciplinary on both diet and physical activity had no impact whatsoever on any of the measurable outcomes after that period of time. By contrast, in Mexico, they put 1 peso on every can of sweetened drinks. And within a year, the reduction had been 6% in the amount of sugar drinks consumed, more than 10% in the poorest groups. And in the subsequent year, it's got even bigger. The effect is even larger on putting a peso on the Coke, but not putting a peso on Diet Coke. And the predicted impact on the Mexican population of that change will be nearly 200,000 fewer type 2 diabetes cases, 20,000 fewer incident strokes. This is not proven yet, but this is modeled from the effects of what's already been seen. A tax bill for the Mexicans saved of nearly $1 billion, and the biggest impacts being in the young people, where it's most economically damaging to get diabetes. So the biggest impact in this from the point of view of societal impact will be, I'm afraid, from fiscal and other measures that change the environment rather than try and convince people to change their own behavior, to make the environment less obviously obesogenic and just make it easier not to do things that make us obese. But of course, I do think alone that won't work, and those individuals who are very susceptible, we're not going to turn to a famine situation, we're not going to become hunter-gatherers again, I doubt. And so we will need to develop cheap, effective, and safe medications, and to do so, we'll need better understanding of the control of human energy balance and the links between obesity and its adverse outcomes. I'd like to finish by thanking my colleagues and collaborators that have worked with me for over many years, physicians who sent us patients, the funders, and of course the patients, participants, and families. And in the last couple of years, we had to add an additional thanks to our thank you slide. So thank you for your attention. I'll be happy to answer any questions. So we have time for questions.
So I don't really understand why there was no effect on the previous study where their food and exercise and everything were controlled, because in a way, I assume they were also consuming less food. So why was there no effect there, and yet they think with the Coke it would make a difference?
So I think when you're dealing with an obese person, that so the thing I've said, diet and exercise do make a big difference to obese people, and they will lose weight. And some of them will keep that off, but a small number, and they'll tend to put it back on. So what I'm not saying is that environment isn't important, but I'm saying that by the time a person has become obese and presented themselves for obesity management, they're already quite far down the line. Now, what we're talking about when we're talking about population changes is we are talking about subtle shifts in many, many, many thousands of people, including people who are only mildly overweight, not becoming obese. We're having impacts across the whole spectrum. And that, I think, is where, we're not saying that changing 1 peso will stop the current obese people being obese. That's not gonna. It's that subtle change at the early phases that is probably having its impact on.
You thought that we were about 20 years
Yes.
to find the right drugs for solving the problem. So what is your opinion a few years later? And second question is, I would like to know, is it more damaging to eat some sugar or take some artificial sweetener?
Okay, so you very good questions, both of them. The first one is, I suppose the question is, are we any better at treating established obesity now than we were 10 years ago? We do have a European licensed drug which has a 10% impact on body weight. That has had profound effects on some people, and so liraglutide at a 3 milligram dose has reduced some people's body weight by 20 kilos, so 25 kilos. So we and it's safe and it doesn't kill you. So some progress has been made in that domain, and other companies and other companies are developing mixed hormones that I've seen preliminary data for that look interesting, and so there's many. So the most reliable approach so far is the gut hormone-based approach, because the problem with the brain as a therapeutic target is that if you use small molecules to target the brain, you can be very effective, but the brain uses the same chemistry somewhere else in the brain for something else. So rimonabant was an anti-cannabinoid drug, was extremely powerful, weight reducing, but it caused severe depression by its action on other parts of the brain. It caused severe depression. The good thing about using natural hormones, in effect, the gut hormones that we know suppress appetite, the good thing about them is that they're natural in a way, and we're just modifying a natural signal, and they're not going to go into other parts of your brain and do other things to your mood or your. So I'm reasonably, I'm still optimistic that by the right dialing, we'll start to get more effective therapies. I am convinced, as I say, I'm not an epidemiologist, and I don't claim deep knowledge of epidemiological methods, but I'm very close to my colleagues in the institute who do this. And Nita Faruhi has done the best systematic analysis on non-sugared beverages, on aspartame and other. And I'm convinced by her data that they are better for you than sugared beverages, and they cause less weight gain and certainly no increased diabetes risk associated with it. And I'm also unconvinced by the cancer scares. I just don't see the data. So I think that although you can have there are lots of theoretical and animal-based reasons why you might not want to use these non-nutritive sweeteners. The actual hard evidence from studies like Interact, which has over 1 million people studied in Europe, is very compelling to me that they certainly don't increase the risk of diabetes if you're a habitual consumer of diet beverages. Whereas if you're a habitual consumer of sugared beverages, your risk is substantially increased. So I think better obviously if you can drink water, then you can have then you have no concerns at all, because nothing will ever emerge. Well, something might emerge. We don't know. If you drink it out of plastic bottles, maybe it's so who knows. But the data is not strong that says that these non-nutrient sweeteners are bad for you. It's not strong data. Jeff.
What can be done to help South Asian people who are insulin resistant without being obese? What can be done to help prevent them developing diabetes?
Well, of course, what you want to do is maintain insulin sensitivity. So as well as exercise is an extremely powerful way of making your muscles more insulin sensitive. So we haven't talked about exercise is not a great way on its own of losing weight, but it's a very good way of maintaining insulin sensitivity. So we wouldn't reduce I would emphasize physical activity and regular aerobic physical activity as a good way of maintaining your muscle insulin sensitivity. There are plenty of trials now of metformin being given to non-diabetic individuals to try and prevent the onset of diabetes and macrovascular disease. There are other insulin sensitizers that are being developed at the moment, which are worthy of trialing. So at the moment, I would say predominantly exercise and possibly metformin as a practical. So thank you for being such an interactive and pleasant audience, and thank you.